Hi everyone, this is our first lecture titled Introduction to Honeybee Biology. In this lecture, we will first have a brief introduction to honeybees and their current health status. This will be brief, however, since we'll delve a lot deeper into the subject later on in the semester. We'll then review some basic biological principles and how they relate to honeybee as a model organism. So first off, honeybees are pretty amazing. If you purposely signed up for this course, you must know this already. But the scientific name of the European honeybee, which is the honeybee that we know, is Apis mellifera. The genus name Apis means bee, and the species name mellifera means honey bearer. So we have a honey bearing bee. Kind of makes sense. When you think about a honeybee, the first thing that probably comes to your mind might be honey, since honeybees produce many things within the hive that we call hive products. There's honey, pollen, wax, something called propolis, which is basically tree sap collected and used to seal the hive up tight, and royal jelly, which we'll also learn about later, just to name a few. Actually, there is such a high demand for honey in the US that we cannot fulfill the demand ourselves. So we actually import large amounts of honey from other countries. Honeybees provide pollination services as well, and as a matter of fact, the value of pollination services by honeybees far outweighs the value of all the hive products combined. You may have thought that honey was where the money was when you're a beekeeper, but actually leasing your bees out to growers is where beekeepers are able to make a living. There are many crops which rely on pollination in order to set fruit. About one third of the US food supply relies on this service. There are exceptions, though. Things like corn and wheat are primarily wind pollinated crops, so they are not reliant on pollinators. If you ever hear that if the bees go extinct, we will too, this is not a true statement. Our diets will just be blander without the many fruits and vegetables and nuts that come from pollination efforts, but we'll still be able to survive. It's estimated that the efforts of pollinators can be valued around $15 billion. A huge issue in the honeybee world, and something you've most likely been seeing in the news, is population declines. Here we have a graph of the number of managed honeybee colonies within the US in millions over time. The red arrows on the graph indicate events that may have affected either the data directly or actual population numbers. The varroa mite, which we will discuss extensively later in the semester, is an invasive mite from Asia that appeared in the US around 1987. It spread rapidly and just wreaked havoc on honeybee colonies, and it still does today. Then in 2006, the term CCD, or colony collapse disorder, was coined. The good news here is you can actually see that the population numbers have risen and now match population numbers we saw in the early 90s. When we discuss the factors that affect honeybee health, there's no one easy answer. Many different issues surrounding honeybee health all interact with one another and make this basically a giant convoluted puzzle that we need to try to put together. I mean, if it were a real tangible puzzle in front of you, it would have no edges, it'd be all the same color, and several pieces would be missing. I can actually feel a few puzzle people out there shuddering. I know I did. But honeybees are faced with all these issues. This includes nutrition, parasites, diseases, pesticides, genetic weakness, queen failure, invasive species, weather patterns, habitat loss, and beekeeper practices. Unfortunately, just to name a few. I will highlight a few of these issues with some detail. The two big players here are parasites and disease and nutritional availability. The varroa mite I mentioned earlier is a very real and very destructive threat to honeybees throughout the US. If you do not have varroa mite presence in your colonies, but your neighbor a few miles down the road does, you will eventually, if not quickly, obtain varroa as well. Worse off, varroa mites are known to transmit about 20 bee viruses as they suck blood from one bee to the next. If you look at the picture on the top there, that is a developing bee. You see the head kind of forming, and a varroa mite. This would be like a mite the size of a dinner plate feeding on us humans. So it's very destructive, very large relative to the body size of a bee. The bottom picture is the best known treatment for a bacterial disease called American foulbrood. Literally burning the hives with the bees inside is the only known way to fully prevent the spread of this. Honeybee inspection services have been developed around this particular disease. That is how bad it is. Nutrition has become a huge issue with honeybee health, with large monocroppings, which are single crop fields. Proper pollinator habitat is becoming more and more scarce. We used to have field borders, full of wildflowers, used to provide some pollinator nourishment, but the need to mow invasive weeds that can take over cropland has basically depleted this landscape. Think about this as how humans need a diverse diet to stay healthy. If we tried to survive on fried chicken alone, we wouldn't be very healthy. Bees also need a diverse diet, but when they're placed in areas where only one or two types of pollen can be collected, they will not do well either. Beekeeping practices are also a big factor in honeybee health. As a beekeeper, you are taking on the responsibility to manage honeybees, which, for all intensive purposes, are viewed as livestock in the agricultural world. This is a big issue with a lack of proper beekeeping education to new beekeepers. Very little funding is put out for extension agents or university appointed educators to go out and educate about beekeeping, so beekeepers are left to their own means of education. Many times, this means a more experienced beekeeper stepping up and educating about their own practices. This can be great in some circumstances, but I have seen beekeepers who pass on detrimental practices to individuals who don't know any better. Some of the time, these practices include not monitoring or treating for varroa mites, which can not only drastically affect the health of your bees, but all bees surrounding you as well. Pesticides are discussed in great extent in the news and the sole reason for honeybee declines, but as we have discussed, it's many factors working together that create these problems. Honeybees and other pollinators may certainly be affected by pesticides if the product is used incorrectly. Most of the time, other pests are being targeted by pesticide treatments, and pollinators, as a non target organism, are also treated. This is being addressed actively throughout the US as we speak, which I will discuss in a second. Colony collapse disorder is a term used to describe a very specific set of symptoms seen in a bee colony. We will talk about these symptoms later, but I wanted to mention that this term is often used incorrectly or used to describe the main issue facing honeybees. A lot of people tend to use the term colony collapse disorder interchangeably with population declines, which is incorrect. I used to work for one of the scientists who helped coin this term, and he said himself that he had not seen signs of CCD for a few years now. There are many more factors, but those are the big ones I wanted to bring up in our introduction. In 2015, the White House put out the National Strategy to Promote the Health of Honeybees and Other Pollinators. This document addressed honeybees, monarch butterflies, and native pollinator species. It had a large focus on habitat availability and nutrition, as well as addressing pesticide issues. This document also requested every state make something called a Managed Pollinator Protection Plan, or MP3 for short. This produced a set of very productive meetings where state officials could meet and discuss pollinator health over a national and statewide level. This also helped stir up discussions on pollinator health and is helping to change public opinion. Whereas bees used to be viewed as, oh, I don't want any of those around because they could sting me. Now it's becoming, I didn't realize how important they were to our food supply. How can I help? There are also many new advocates for honeybee and other pollinator health. This may even include you for signing up for this course. So, all in all, the bleak picture that the media tends to paint is not really all that bad in the pollinator world. There's definitely things for us to improve upon, but we're working at it every day. Now that we have the overview covered, we can focus on this course specifically and what my expectations are for you. There will be many new concepts to present and entomological language to learn, so there will be a certain amount of memorization involved. My hope is that you can move past just the remembering stage and utilize this new information and move closer to the top levels of this graphic here when you start applying and analyzing the information on your own. This course will use the honeybee as a model organism, as it is very widely studied. and will be an interesting mix of basic bio and then using these concepts when we dive into honeybees specifically. We have a large mix of students in this course who greatly range in their science background, so keep that in mind as we progress, as I will try to provide great detail where needed. So let's look at specific characteristics of life. We have cells and organization, energy use and metabolism, growth and development, reproduction, regulation and homeostasis, evolution, and response to the environment. Focusing on cells and organization, organization can refer to the differentiation between the internal and external environment. Cell theory states that all organisms are made up of cells. Cells are the smallest unit of life, and cells come from pre-existing cells. We have prokaryotes, which include bacteria. These are generally single-celled organisms with a more simplistic cell structure than eukaryotes. Eukaryotes include plants, animals, yeasts, and fungi. These may be unicellular or multicellular organisms, generally with a more complex cell structure. Looking at energy use and metabolism, all life uses energy from the environment to maintain order. Plants need sunlight, water, and other compounds to survive. We, as animals, need to eat, drink, and breathe to survive. All life breaks down and synthesizes chemicals. Some examples of this include, but are obviously not limited to, the release of energy, known as respiration, or maybe the production of essential components, known as anabolism. Then we have response to environment. All organisms respond to changes in some fashion. There are so many examples of this, it would be really hard to list any kind of majority. I'll just mention hibernation, where organisms adjust their cellular activity depending on the abundance of nutrients available. And plant growth toward the light is another great example. I can always tell when I forgot to turn the plants in my office window since they'll be standing at a 45-degree angle instead of straight up. But other examples could be things like migration, defense, even sweating, hunger, or blinking, for example. Next is regulation and homeostasis. So, what is homeostasis? It is an organism's tendency toward achieving a relatively stable equilibrium between its interdependent composing units, especially as maintained by various physiological processes. You got all that? Well, less formally, it is maintaining internal stability in response to environmental changes. So a good example would be maintaining a stable internal environment or regulating body temperature. Highly simplified, this is completed through chemical signaling in the body. This figure shows an example of how many interacting parts are involved for this maintenance at any single moment in the human body. There is reproduction. All living organisms have a finite lifespan. Death is imminent and will occur in an organism's lifetime. Individuals pass on genes through reproduction. This tends to be a main goal of nature to make as many viable offspring as possible. DNA contains information by which an organism transmits structure, development, and function to its offspring during reproduction. Now, growth and development. Organisms add cells to themselves and cells grow larger. Cells, organism, tissue, organs, etc. change in state over time. Like this image of a plant producing roots, stems, and leaves. You can see how much larger it becomes from this process. Kind of like myself after the holiday season. Finally, we have evolution. Organisms make imperfect copies of themselves due to random genetic mutation. This change occurs over time, usually long amounts of time and multiple generations. different organisms interact with the environment differently. This allows for adaptation and specialization, in which we will discuss evolution a bit further next lecture. So just to see them once more, here are the characteristics we just reviewed. Taking these principles and looking at them in regards to levels of biological organization helps to see how many different scales these interactions occur within. The smallest level, which is not a unit of life, is an atom, which bond to make molecules and macromolecules. Then we have cells, tissues, organs, organisms, populations, communities, ecosystems, and then biospheres. So when we discuss biological levels, there's a big variety there that could be looked at. Next comes some scientific terminology. Hypotheses, or a hypothesis, this is a scientifically testable prediction based on observations. Hypotheses must make testable predictions that can be shown correct or incorrect. Additional observations or experiments support or reject a hypothesis. A hypothesis is never really 100% proven. I remember learning about the scientific process and how one of my professors would correct you anytime you stated that the data proved a prediction to be true or false. Nothing in science can actually be proven, he would say. That really stuck with me. Hypotheses can be rejected over time. A good example of this is the first envisioned models of the universe. Ptolemy's geocentric model showed planets, the sun, moon, and stars orbiting Earth. This was eventually rejected as Copernicus stepped in with his heliocentric model, where the sun and stars were stationary bodies in the universe, and the Earth orbited the sun and the moon orbited the Earth. This was also rejected over time as we learned that the sun and stars are not stationary. So, what is a theory? It is a broad explanation of some aspect of the natural world that is substantiated by a large body of evidence. Theories come about as the result of a large collection of hypotheses repeatedly tested to be valid. This also allows us to make predictions, and though it can never be proven true, it is extremely likely to be true. Examples of scientific theories include Einstein's theory of relativity or Darwin's theory of evolution. Now on to laws. So, what is a law? It is a description of what nature does under certain conditions. These are statements in which no exceptions have been found. These are accepted to be true and universal, but they are rare. Newton's law of gravity is a good example. No one has found any evidence to refute the existence of gravity that I know of. We also cannot escape this lecture without the mention of the scientific method. Now, personally, I think every time I was presented the scientific method, it was slightly different. This one might be different from what you may have learned in the past as well, but know that regardless of how the steps are listed, they basically have the same result. So we'll just stick with this guy here. So first is state the problem. This is where a question is made regarding a natural phenomenon observed. Second is make observations. This is collecting background information regarding the system in question. Third is forming a hypothesis. This is where background observations lead to forming a hypothesis and making predictions that will try to explain the phenomenon in a testable way. Fourth is test the hypothesis. Experimentation is conducted here and data analyzed to determine if the predictions of the hypothesis are correct. Lastly, and fifth, is draw a conclusion. This is your end step where you determine whether the hypothesis is accepted or rejected. When in the fourth step, test the hypothesis, it is necessary that we discuss controls and analyses. Data are often collected in parallel with a control versus experimental group. These groups differ by only one factor, which we call the independent variable. The control group and experimental group's data are used in the data analysis. You are looking for something called a statistically significant result. What this means is you can determine if the control and experimental samples are likely to be different from each other because of that single variable that is different between the two samples. If the comparison between the control and experimental groups are not statistically significant, then you cannot support or reject, depending on the situation, your hypothesis with any certainty. Here's just a practical example of the scientific method steps here. We are going to state the problem from natural observations. These are steps 1 and 2. Our observations here are that leaves on maple trees fall in autumn when the days get colder and shorter. Next is forming a hypothesis, or our step 3. So our prediction is that a shorter amount of daylight causes the leaves to fall. Next is testing the hypothesis through experimentation, or our step 4. In this experiment, small maple trees are grown in 2 greenhouses where the only variable is length of light. The control group, which is basically the unadjusted group, gets a consistent amount of daily light, and the experimental group, which is the adjusted, gets a progressively shorter amount of daily light. The data analysis here is actually included in our step 4, and we can clearly see from the graph of the number of leaves dropped per tree over group type that the experimental group dropped a greater quantity of leaves than the control group. A statistical test would be needed to determine if the results are statistically different or significant, so we can accept or reject the hypothesis, but in this case they are statistically significant. The final step is to draw a conclusion, and here we can accept our hypothesis that a shorter amount of daylight causes leaves to fall. I also wanted to give a brief mention to the structure of scientific literature, as we will be reviewing some scientific papers throughout the semester. At first, these papers can seem overwhelming or confusing as to how they're structured, but hopefully with this overview, it will help you understand which sections contain what information in regards to the scientific method. Scientific papers actually do follow the scientific method in the way that they report their findings. The section called the Introduction contains steps 1 through 3, or stating the problem, making observations, and forming the hypothesis. This is where the background information for the study is found. The Materials and Methods section contains step 4, or testing the hypothesis. This is where the researcher or researchers let you know how they did their experimentation. The Results section also contains step 4, testing the hypothesis. This is where the results are shown in any statistical testing. The Discussion Conclusion section contains step 5, which is drawing a conclusion. This is where the researcher discussed what their results mean in the context of their study. It is also where they will reject or accept their hypothesis. Just fair warning though, it's not always as cut and dry as it seems. Just to walk you through an actual example of what a scientific paper looks like, in which I know that it's really hard to read this whole paper on one slide, so you don't have to worry about the actual details here, just I'll point out sections. But here's the first page of this particular publication. First, we'll look at something called the abstract, which I didn't mention before, to try to reduce confusion, basically. But this section of the paper is a shortened summary of the entire scientific method. It contains a small amount of information from each of the steps we reviewed. The reviewer will summarize the entire study in the most condensed way possible, and you should be able to draw conclusions really just from this section. Next, we will see the introduction, which again is steps 1 through 3, and the materials and methods section, showing step 4. The next page has the results with the remainder of step 4. And finally, the discussion with step 5 information. Hopefully that wasn't a massively overwhelming amount of information for your first lecture. Thanks for hanging in there, and I look forward to our next lecture.